Thank you, Amy, for kicking us off in real-world evidence, real-world data, and some criteria that Flatiron, especially since Amy joined, have derived. I feel in this particular instance between Amy and our next speaker, Gaurav, Singal, as a bridge between the same company, because as you all know, Flatiron is now owned by Roche, and Foundation Medicine is as well. So we're very pleased at the CEO Roundtable to welcome Dan O'Day as the newest member of the CEO Roundtable this November. And now Gaurav is going to take us really into actually assays and biomarkers and the importance, essential importance that they play in patient selection. So please, Gaurav.
Thanks, Marty, for the introduction, and thanks for the invitation to be here. I'm thrilled to be able to share some of my thoughts on this topic with you. Less thrilled to follow Amy because she's such an incredible speaker. And I'll cover some of the same material she did, but I'll do so quickly. So as far as disclosures, I'm an employee and shareholder of Foundation Medicine. I'm hoping to talk to you about a few things today. First, a little bit of background and context on biomarkers and how they relate to real-world data. Frankly, I thought yesterday was an exceptional series of discussions, and I think many of the challenges that we need to tackle as a community were addressed there, so I'll highlight this pretty quickly. I'll describe for you, similar to Marty, a case study in how we can bridge at least two, I think, important data silos in real-world data, being clinical data and genomic data. Again, sort of focused on the world that I inhabit, but one that I think is generalizable to the field. And then finally, I'll share a couple of examples, some of which Amy has touched on, some of which I'll add on where some of these capabilities might actually impact some of the challenges that we've all identified and hope to address, I think, together. So quickly, by way of background, again, I think some phenomenal discussions yesterday by many of our speakers. We know that biomarkers are critical for the success of immunotherapy and on which patients should get therapy and should not, or maybe should get a different therapy or combination. But we also know that there's several reasons that we're not there yet. One of those reasons is the complexity. I think many of us believe, and was evidenced yesterday, there's not a single biomarker that's probably going to sufficiently predict those who will and won't respond. The data that we have largely have been generated through clinical trials, and I think Emmett brought this up yesterday in his talk, but many others did as well. We have a series of studies that have each explored the question in their own particular way, and how we look across them is non-trivial. And then finally, and Amy's touched on this a fair amount, I'll extend this a little bit into the biomarker world. It's hard to generalize sometimes from the data that's collected in a controlled setting to the broader population that all of us clinicians end up treating. We've also heard a lot about this from Amy, so I won't dwell on this, but we've seen real-world data emerge in a really powerful way recently. Obviously, it's been going on for a long time in the academic medical world. The notion of studying and learning from observations of clinical care and practice. These are just a few of the folks that were here and is obviously not comprehensive. We've also heard of a number of consortia. Amy mentioned these in one of her last slides. I'll call out Stand Up to Cancer specifically because it came up a couple of times yesterday and has really pushed the field forward in many ways, but obviously there are many other consortia. And then I'll touch on one specific industry collaboration, but as you saw from the previous slide, there are a number of folks sort of tackling this challenge. We also heard from Amy about some of the work that FOCR has led and a policy framework and white paper that just came out very recently that highlighted some of the things that Amy's already touched on. So let me spend the bulk of the time telling you about one specific example of how we can bridge data silos, and this is a perspective from, again, two what were historically independent private efforts, as Marty pointed out, have come together or are coming together more closely now. But the work that I'll describe here, Amy and I worked on together, started about 3 years ago, and the challenge that we sort of had taken on is each of us independently had the potential to impact the field through our individual data sets that in some ways represent in their own way an implicit data sharing. I'll talk about that in a second. But how could we bring those two data sets together to create the data set that I think many of us in the oncology world were hoping for, which is a linked clinicogenomic data set. So briefly on the two data sets, I'll describe the Foundation Medicine data set first, and then Amy's already talked a bit about the Flatiron side. Foundation Medicine, for those who don't know, sequences patients with cancer by working with providers all over the country and all over the world. We serve hundreds of medical practices and centers, and we sequence now a little over 2,000 samples every single week. Implicitly then, we've become a clearinghouse for all of these centers and the genomic samples that are processed, or the tumor samples that are processed and sequenced. And implicitly then, we've enabled a type of data sharing, or at least data integration, for genomic data by virtue of the cases that we've sequenced for all of these otherwise independent and disparate centers. Those sequencing results are obviously returned back to the individual providers in those centers in the form of reports that are used to take care of patients. But de-identified data from each of those sequences is aggregated for the purposes of research, and that includes genomic results, tumor mutation burden, microsatellite instability, and for many of these cases also PD-L1 testing. So many of the sort of core components as we think about immunotherapy that we may want to study at scale beyond what we're able to see through clinical trials. On the Flatiron side, you've seen the slide from Amy as well. I'll just draw the parallel that in the same way that in some ways Foundation Medicine is integrating genomic data from disparate individual institutions and centers, Flatiron is essentially doing the same thing on the clinical data side. And again, we've quite a bit of detail on that front. So here's the tough part. How do you bring those two things together? And I put this slide up not because I intend to walk through it in detail or that folks in the back can even necessarily read it, but just to highlight that this is not a trivial problem to solve. And actually many of the first several months, if not the first year of our partnership, was spent on trying to solve this problem. And the problem was essentially this. Independently, we each have data that has PHI only in the context of serving the patients for their clinical care. Our research data sets are all fully de-identified. So how then do you bring a de-identified data set from one institution or one entity together with a de-identified data set from another entity and preserve privacy throughout that whole process? So the way we approach this, and we've written about this and published it, is basically we went through a third-party independent clearinghouse, but who also could not see PHI. And so the way the problem was ultimately solved is this third party provided a tokenizing engine that took the PHI for each of our respective institutions within our entities and scrambled them, but scrambled them in a deterministic way. So the same PHI from one of our organizations became the same scrambled 36-digit code, which could not be then interpreted to get back to the PHI, but which could be used to then link those records through this third party. So we ran that engine on each of our records independently. Those then scrambled codes were sent to this third party that performed the linking. They sent back then the overlap or crosswalk between the two data sets, and that allowed us to then merge fully de-identified data in a fully compliant way and create what ultimately has become the linked clinical genomic database. And I share this because it wasn't a trivial problem to solve. I think in the spirit of Amy's comments that many of the sort of opportunities we have as a community to share is really on the how and the methods. And this was a really important part of the sort of methodological approach that we spent a lot of time trying to figure out. So where did that bring us? This is actually a moving target because we relink these data on a quarterly basis. At a recent link, this was over 30,000 cases. I think it's now about 35,000 cases, and again continues to evolve. I think a year ago, similar to Amy, when I shared these numbers, it was half this size. And those 30,000 cases are pretty remarkable because they contain the full genomic data, alterations, tumor mutation burden, MSI, and then all the bioinformatic sort of below the surface information about all of those findings, and on the clinical side, the full depth and sort of rigor of the clinical data that we've heard about from Amy. And just as a snapshot of what this means from a tumor by tumor perspective, and again, these data I will say are approximate because every month this moves, every quarter this moves, but roughly you get to see the order of magnitude of these data sets, and then there's a long tail of other tumor types, as you all are quite aware. So this slide, again, is not intended to be dissected, and again, my goal here is really to give an overview, but it was really important for us as collaborators in sort of bringing together now two disparate entities to approach this with the same level of rigor and validation that you've heard from Amy on the clinical data side. And so we did this validation work last year and presented it at ASCO, and it's currently under review as a manuscript. But basically, we did a few things. Number one, we looked at the baseline characteristics, and we said, do the patients in this data set represent and reflect real-world patient populations in oncology. And the sort of short version of it is that in both genomics and clinical characteristics, they're highly representative with the slight skew driven by which patients are getting sequenced. And those patients tend to be patients who have either been treated multiple times or tend to be the sort of non-traditional young non-smokers in non-small cell lung cancer. Their genomic characteristics compared to TCGA are also very representative, with the caveat being that these are potentially later stage patients who have gone through now multiple lines of therapy. Next, we looked at known prognostic implications of both clinical and genomic factors that have been well established through the literature already. And we confirmed that these genomic and clinical characteristics in the data set did indeed confer those same prognostic implications that have been previously documented. And then finally, we looked at the implications for genomics and clinical characteristics on treatment and response. Again, looking to recapitulate findings that are well established in the field, things like response of EGFR inhibitors in the presence of an EGFR mutation, response to NCCN-listed therapies in the presence of NCCN mutations, and then finally, concordance and discordance between NGS-based results and single biomarker FISH PCR-based results, IHC-based results, and their implications for clinical care. Long story short, this was an important piece of the first step in what will be an evolving sort of effort to determine where exactly one can reliably use real-world data, collected, integrated, linked in a somewhat non-traditional or novel way, because it's important that we not sort of overextend the ability of real-world data and that we sort of take those steps in a measured way. And this was an important first step in that process. So that's sort of one example of a data set that's been created to integrate clinical genomic data. I think, as Amy alluded, there's many other efforts that have similar aspirations. I want to spend the next few minutes talking about what we might be able to do as a community as these data sets become mature. So we talked about one problem in the field of generalizability. Amy's touched on the left side of this quite a bit around the notion of the patients that ultimately make it into clinical trial. I'll highlight a second one that I believe Rich commented on in his presentation yesterday on the challenges of tumor-agnostic approvals. So if you look on the right slide, this was a table that I believe Rich included in his presentation of the pembrolizumab approval in pan-tumor MSI. And as Rich pointed out, there's a reasonable distribution and representation of tumors, but this is a really rare finding, right? Especially outside of colorectal cancer and some of the other more prevalent tumor types. And the notion of finding patients who are MSI high and treating them with cancer immunotherapy in the context of a clinical trial can only be so expansive if we want that trial to be sort of reasonable in terms of cost and duration. And on the one hand, I think as the FDA pointed out yesterday, there's a desire to make these therapies available broadly for patients who might benefit, and that has to be balanced with the sort of burden of evidence collection, and that's a tricky balance. So how can real-world data solve that? You saw this from Amy on the sort of entire clinical data set. The clinical genomic data set similarly has a representative population of patients over the age of 65 who actually comprise more than half of the patients who got immunotherapy in this linked data set. But then I want to touch briefly on the MSI example because that I actually think is pretty remarkable. This is work that our two organizations did and presented at the ASCO SITC conference. Actually, I believe this was last year. And the sort of takeaway from this is, in this linked data set, if you look across all of the patients, across all tumor types, you see, and this was data back from May of 2017, so right around the time of the pembrolizumab approval for pan-tumor MSI high patients. And you see the sort of bottom right figure with blue and green bars. Blue represents all of the patients who are MSI high across the entire cohort, and green represents the subset of those who had been treated with pembrolizumab. So this was a year ago, just sort of peri approval of pembrolizumab in that indication. I share this because not because these data necessarily help us answer that question, but this shows the potential of a data set like this as it extends. And even if we were to relook at these data now, we would likely see a lot more use of cancer immunotherapies across disparate tumor types in the context of microsatellite instability. And we might be able to extend some of the findings of that initial trial that led to approval and understand both tumor types that maybe not been represented in that initial study, or maybe that were not represented sufficiently just by function of rarity. Second, I want to comment briefly on heterogeneity. So these are just two recent New England Journal articles about important investigations of combination therapy, which is obviously the topic of this forum. And obviously the therapies were different across these, but even the biomarkers studied. One used tumor mutation burden, one didn't, used a specific cutoff of 10 mutations per megabase, used multiple different thresholds for PD-L1. And as you can imagine, sort of looking at these data sets or results across different studies becomes a non-trivial exercise. I borrowed this slide from Emmett. Hopefully he doesn't mind. I took a picture during his presentation, and I wanted to remind the audience of this slide because I think it wasn't your slide. Sorry, you get credit for it anyway, I guess. Apologies to whoever's slide it actually was. Point being, I think that if you look across all these different studies of different agents, they look at both different methods of measuring tumor mutation burden and also different cutoffs. And so I think an important question becomes, how would we ever approach standardization or even understanding the role of tumor mutation burden across different agents in monotherapy, let alone combination therapy? So this I'll caveat with the same caveats that Amy provided, which is that these haven't been validated by a statistician. There are known confounders in these data, and the goal here is not to actually share a scientific result, but rather share a capability and an illustrative possibility. So this is just a simple application that was built on top of the data set by one of our data scientists that looks at a number of different cancer immunotherapies in this linked data set, and you can see those listed on the left here, looks at a common measure of tumor mutation burden, all measured on a single platform, and then allows a couple of different modifications. And what's plotted on the right is survival. And what I'll show you here now is just a quick video of how the survival curves change as you change the stratification level by TMB. And as you increase the TMB threshold that you're using to stratify the two curves, you can see the evolution or sort of the dynamic of survival. So let me just see if I can play that for you here. So red is the quote-unquote TMB high. And you can see as this TMB threshold increases, you see sort of almost a dose-dependent separation of those curves. So again, lots of confounders here that aren't accounted for, and this is not meant to be a scientific endpoint, but only to show that here's an opportunity for us to look across multiple cancer immunotherapy agents in a single data set with a single measure of tumor mutation burden with sufficient scale that we might actually be able to derive meaningful results, all with clinical endpoints that have been curated in a harmonized way across hundreds of centers across the country. That alone, I think, is a pretty remarkable capability that I think may unlock a lot of things for the field and for many of us in this room. And I should also note that we could do this sort of individually by therapy as well and see if the same sort of results hold across different therapies. Finally, I'll make a comment on complexity and specifically on resistance. And as we think about combination therapies, obviously understanding primary and secondary mechanisms of resistance will be really important in that study. We heard a lot about that yesterday as well. I want to highlight the role that real-world data has already played in this. Real-world data maybe in the more traditional sense. So first, the article on the left, which I'm sure many of you know, remarkably the result of a real-world observation of 4 patients at a single center, yielding, I think, some of the earliest insights into mechanisms of resistance to PD-1 blockade. And on the right, I think you see the power of collaboration, and Stand Up for Cancer was an important part of this effort, along with a number of other academic collaborators in pooling sort of clinical observations, again, from the real world, in understanding additional mechanisms of resistance. And I share these, again, both because real-world data in some ways is not a new concept, right? Clinical centers have been using real-world data and observation, and a lot of the field has advanced because of that. I think the conversation really here is how can we scale all of those efforts that actually many of us are quite familiar with. So I'll conclude with just a few thoughts. One, that real-world data sets are emerging in a level of maturity and scale that I think is new. And that's across not only academic medical centers and consortia, but also through industry-driven initiatives. That these data sets, I think, pretty clearly can complement and extend data and evidence that's generated through clinical trials. They're not a replacement. I think many of us acknowledge that. But there are real purposes that they can serve. And there are additional applications like trial design, post-approval efficacy, which Amy touched on. And I think this especially may become relevant in the world of histology-agnostic therapies and approvals, as, again, Rich talked about yesterday, largely because they will be really hard to study with the same level of scale that we would for a more sort of narrowly defined population. But there are a lot of open questions, I think, for us to tackle as a community and from a policy perspective. And Amy touched on these a little bit already. Can we accelerate the development of some of these challenging therapies from an R&D standpoint for niche patient populations by using external real-world control arms? Can these data be used to actually facilitate discovery in a way that real-world data is not traditionally, I think, thought about? And part of that will probably require more creative ways of engaging patients in research in the context of real-world data. I know it's something that many folks in this room think about. And then finally, a topic that has not really been a core part of our discussion here, but I think is one that will become increasingly relevant, is as biomarkers and sort of the methods of stratifying patient response and selecting therapy become more and more complex, and as there are not only multiple monotherapies but multiple combination therapies to select from, the challenge of being a clinician will become exponentially harder as well. And if I put on my clinical hat, having to figure out exactly what combinations of biomarkers I need to test and how I interpret all of those to select the best therapy for my patient, I would suggest that data and technology are likely to play a really important role in actually enabling the bridge of complexity that will be required to actually translate this into clinical practice. So with that, I just want to say thanks again for the opportunity to be here and share my thoughts and for a lively discussion.